Well, good afternoon. I want to welcome you to the soft tissue sarcoma breakout session. We're really delighted to have a stellar group of expert physicians to talk to us today and bring us updates about soft tissue sarcoma. Each of the speakers will have about 45 minutes, and we hope to entertain any questions that you have at the end of each of the presentations, as opposed to like this morning holding to the end. So I would like to introduce all of our speakers for today. We have Professor Robin Jones, who will start us off. He's a medical oncologist at the Royal Marsden. He's head of the sarcoma unit, and he specializes in soft tissue and bone sarcomas, as well as in phase 1, phase 2 clinical research. He will be joining us online. And so we have Dr. Peter Reichert, who's here with us in the room. He is a medical oncologist at Helios Hospital, Berlin-Buch. He's head of the Sarcoma Cancer Center. He also specializes in the care and treatment of soft tissue sarcoma and bone sarcomas. And then we will have a break, and after the break, Professor Annette van der Graaf will be speaking with us, and she's a medical oncologist at Antonie van Leeuwenhoek and the Netherlands Cancer Institute. She is the president of the EORTC, and she specializes in soft tissue sarcoma, as well as has a special interest in adolescent young adults with sarcomas. So I think you're in for a treat and really a wonderful update on soft tissue sarcomas. So we'll now turn to Professor Jones, I believe is online. And he will
Hi,
be
yes,
joining
can
us.
you hear me?
Yes, we can hear you.
How are
We're
you?
just switching the screen so that we can actually see you as well. So it'll just be 1 minute.
No problem. I'll try and share my screen as well.
We see you now.
Oh,
Hello.
fantastic. Hi, good to see you. Can you see my slides?
We can see your slides, yes.
Oh, wonderful. Well, thank you for the kind invitation. It's a real pleasure to be with you. I wish I could have joined you face to face, but unfortunately, this time it was not possible. Thanks for the kind introduction, Denise, and I'll just switch into presentation mode. So it's been very busy in the UK, plus there have been rail strikes which have made travel very, very difficult. So unfortunately, I think many of the UK participants were unable to attend in person. These are my disclosures. And this is a plan for the lecture. I think I've got too many slides, so Denise, you'll have to stop me at some point, but I'll try and go through this quite quickly so that we have more time for questions at the end. I'd like to split this into localized disease and then advanced disease. In the localized disease setting, just go over some of the recent data regarding the use of chemotherapy in the early setting in terms of soft tissue sarcomas, and in a way, a resurgence in the use of neoadjuvant chemotherapy. And in the advanced disease setting, I'd like to highlight some of the recent drug approvals, which is clearly a very positive thing for patients and oncologists, particularly in GIST with the approval of ripretinib and avapritinib, but also the approval of NTRK inhibitors and tazemetostat in epithelioid sarcoma, as well as ongoing trials of promising new agents in other subtypes. So as I said, I've got a lot of slides, but that I think is a very good thing and a reflection that there's a lot going on. And then of course, finally, I'd like to finish with a conclusion. So in terms of localized disease, historically, there's been a lot of controversy regarding the use of pre or post-operative chemotherapy for the majority of soft tissue sarcomas. I would say that over the last 5 or 6 years, there has been a resurgence of interest and use of neoadjuvant chemotherapy. Of course, the Italian, French, Spanish, and Polish randomized trial, I think, has contributed to this. So a trial of anthracycline plus ifosfamide chemotherapy or standard chemotherapy versus chemotherapy tailored to histological subtype led by Alex Kroenke. The inclusion criteria for this trial were very stringent compared to a lot of previous trials performed over the last few decades. So it included patients with localized soft tissue sarcomas of the extremity and trunk. Patients could have primary or locally recurrent tumors, but importantly, the tumors had to be high grade, had to be larger than 5 centimeters, and had to be deeply seated. The primary endpoint of this trial was relapse-free survival, and secondary endpoints included overall survival, radiological response, and the response in the pathological specimen at the time of surgery. And this slide outlines the randomization. So people were either allocated to receive standard chemotherapy, so the epirubicin and ifosfamide treatment for 3 cycles, or to receive histology-tailored chemotherapy. So for patients with myxoid liposarcoma, that was trabectedin. For leiomyosarcoma, that was gemcitabine and dacarbazine. For pleomorphic sarcoma, that was gemcitabine and taxotere. For synovial sarcoma, that was high-dose ifosfamide. And for malignant peripheral nerve sheath tumor, that was ifosfamide and etoposide. And the bottom line from this is that the trial initially showed a significant benefit in terms of relapse-free survival for the standard chemotherapy arms, so the epirubicin and ifosfamide, as shown on this Kaplan-Meier curve. It also showed a benefit for the standard chemotherapy arm in terms of overall survival, as shown on this Kaplan-Meier curve. So they have published updated results of this randomized trial in 2020. And with the updated analysis, the advantage in terms of disease-free survival became less, but the advantage in terms of overall survival is maintained. Further work by Sandro Pasquale and colleagues have used a circulator nomogram for predicting 5 to 10-year overall survival post-surgery based on a number of factors, including patient age, the type of tumor, the size, and the grade. And they've applied this predictive tool to the largest adjuvant chemotherapy or post-operative chemotherapy trial to see whether the circulator nomogram can predict who would benefit from post-operative or pre-operative chemotherapy. And in their analysis, they showed that post-operative chemotherapy halved the risk of death in patients with low predicted overall survival. So again, further evidence supporting the use of anthracycline plus ifosfamide chemotherapy, either in the pre or post-operative setting for high-risk soft tissue sarcomas. And I'd say that this, that all of these pieces of information have contributed to a more solid evidence base for the use of chemotherapy in the localized disease setting, and to have discussions regarding the pros and cons of giving such treatment with individual patients. Of course, the chemotherapy drugs do have potential long-term side effects, such as heart damage, kidney damage, as well as the potential for causing secondary cancers. But the risks of these are fairly low. But it is important to have a very straight discussion with people regarding the pros and cons of chemotherapy. And the information that I've just presented, I think, is very helpful in this regard. Another important point to make is that clinical trials are essential in order to improve our understanding and improve the outcome for patients. And of course, the EORTC are currently running the randomized STRAS-2 trial for retroperitoneal liposarcomas and retroperitoneal leiomyosarcomas. And the design of the trial is shown on this slide. So people are allocated to either receive surgery alone or to receive preoperative or neoadjuvant chemotherapy before surgery. And for liposarcomas, this is 3 cycles of doxorubicin and ifosfamide. And for leiomyosarcomas, This is 3 cycles of doxorubicin and dacarbazine. So a very important trial currently recruiting patients all over Europe. Gaining a better understanding of the biology of these tumours is also very, very important, and we've set up a sarcoma accelerator network to try and link the clinical researchers with biological researchers, with a grant to the UK, Spanish and Italian authorities. And basically, this ambitious proposal, this ambitious grant seeks to better define the role of chemotherapy in localized soft tissue sarcomas by collecting various pieces of information. So not only collecting clinical, radiological and pathological data, but also collecting multi-omic data from genomic and epigenomic profiling and proteomic profiling, but also functionally assessing these things in preclinical models, including patient-derived models, immune-competent models, and also performing functional genomics. And then finally, in the 4th program, trying to bring all of this together to develop other predictive nomograms of response and prognosis, as well as developing artificial intelligence pipelines for integrative analysis of all of these data, and mathematically modeling mechanisms of response and resistance. And tissue will be collected serially during treatment, so pre-treatment, post-treatment, and if relapse occurs. And this is built into recruiting patients into the STRAS-2 trial as well. So moving on to advanced disease. Of course, huge progress has been made in the treatment of metastatic gastrointestinal stromal tumor. And I know there's a parallel session on this. Of course, imatinib has revolutionized the treatment of advanced disease. Sunitinib and regorafenib have previously been approved based on randomized placebo-controlled trials. But I'd like to just quickly mention ripretinib and avapritinib, which have been approved more recently. So ripretinib showed promise in a phase 1 trial, and this phase 3 trial randomized patients 2-to-1 to receive either ripretinib or placebo. And patients had previously been treated with at least 3 prior lines of treatment. And the primary endpoint of this trial was progression-free survival. Secondary endpoints included response rate and overall survival. And patients allocated to placebo could cross over to ripretinib on progression. And patients with progressive disease on ripretinib 150 milligram per day could go on to 150 milligram twice a day on progression. So the baseline characteristics of all the people entered into the trial are shown in on this slide. There was very well balanced in terms of the clinical and molecular characteristics of the tumor. And the trial met its primary endpoint with a very impressive improvement in median progression-free survival of 6.3 months for ripretinib versus 1 month for placebo, with a hazard ratio of 0.15, so a very statistically significant result. And similarly for overall survival, so median overall survival 15.1 months for the ripretinib arm. In terms of tolerability, the drug is relatively well tolerated. Notable side effects can include alopecia, which is unusual for this type of drug, but also laboratory abnormalities. But I'd say that in general, particularly compared to sunitinib and regorafenib, this is a well-tolerated drug. So avapritinib, again, has been approved by the FDA based on the results of the phase 1 trials that we performed, basically looking at 2 populations, one with patients who had tumors harboring the PDGFR alpha exon 18 mutations, particularly the D842V mutation, which is resistant to all other tyrosine kinase inhibitors. And then for patients who'd received at least 3 lines of prior treatment for metastatic GIST. And there were 43 evaluable for response in the PDGFR alpha group, and 111 in the 4th line group. And again, a typical sort of clinical and molecular characteristics for this patient population. And of course, the drug was very, very active in patients with exon 18 PDGFR alpha mutations, with a 86% overall response rate, as shown on this slide. And very durable responses as well in this population. So a very active drug in this group. I think one of the most important things to bear in mind in terms of the side effects of this particular drug is the risk of neurocognitive toxicity. And it's important to be vigilant for this and to stop the drug and adjust the dose if there is any sign of loss of concentration, loss of focus, poor memory. Very, very important to be vigilant for this side effect. And in this trial, 8.3% of patients discontinued avapritinib for treatment-related toxicity, 2% discontinued treatment for cognitive effects. And just to show how this can really have an impact on a patient's quality of life, this is a patient who's referred for the phase 1 trial, a gentleman in his 40s who had been previously treated with imatinib, sunitinib, and regorafenib. As you can see, when he was referred to us, he had a huge tumor, as illustrated on this slide, and had a dramatic response on avapritinib, and actually went on to have surgical resection of the residual tumor. And he continues to do well. He's actually had a more recent scan than this one from April of last year, and remains disease-free. So a very effective drug in this patient population. Moving on to other types of sarcoma, we're clearly moving away from using just anthracycline and ifosfamide in sarcomas. There are a number of subtypes where we would use different drugs for different diseases. But of course, for the majority of patients, the mainstay remains anthracycline drugs with or without ifosfamide. There are a number of second-line treatments available, so gemcitabine and docetaxel, gemcitabine and dacarbazine, pazopanib, trabectedin, and eribulin. And it's, again, very pleasing to see that over the last 10 years or so, pazopanib, trabectedin, and eribulin have been approved by the FDA. This is just an overview of some of the randomized trials over the last 10 years. Particularly in the first-line setting, where a number of trials have randomized people to receive either doxorubicin alone or doxorubicin in combination with another agent or another combination therapy. And from all of these, none of them have shown an overall survival benefit for a combination over doxorubicin. Pazopanib, of course, was approved based on the Palette trial, pazopanib versus placebo, showing a benefit in terms of progression-free survival for pazopanib. And similarly, trabectedin was approved by the FDA based on a randomized trial in lipo and leiomyosarcoma, randomizing people to receive trabectedin or dacarbazine. And similarly for eribulin, as shown on that bottom box. There are other exciting developments in terms of treating metastatic disease. The FDA have approved the EZH2 inhibitor tazemetostat in epithelioid sarcoma. I'll mention that in a few slides' time. also NTRK inhibitors, but I suppose the major question that remains there is which subtypes do we need to specifically focus on in terms of NTRK rearrangements. The EORTC have performed a trial of an ALK inhibitor in inflammatory myofibroblastic tumor. Immunotherapy in the form of checkpoint inhibitors has shown efficacy in angiosarcomas, potentially in pleomorphic sarcomas, and a very sensitive tumor type to checkpoint inhibitors is alveolar soft part sarcoma. Certain T-cell therapies have shown promise, particularly in synovial sarcoma and myxoid liposarcoma. And of course, CSF1 inhibitors have shown activity in tenosynovial giant cell tumor, as well as several other drugs in desmoid tumor. So NTRK inhibitors. So these have an essential role in nervous system activation. Gene fusions in these can induce proliferation and engage downstream signaling pathways. These abnormalities are rare, but they can occur in a diverse range of tumors, including sarcomas. And that just shows a slide of TRK receptor signaling and all the downstream pathways. There have been a number of successful trials of various NTRK inhibitors in a number of different cancer types. This is the publication of larotrectinib in adults and children with tumors with TRK fusions. And similarly, this is the publication for entrectinib in patients with NTRK fusion positive solid tumors. In terms of the sarcoma patients, there have been very impressive response rates in sarcomas with TRK fusion sarcomas, as shown in this slide. And again, durable responses. And this can have a huge, again, a huge impact on a patient's quality of life. This is a patient that I've been treating, a gentleman in his 40s, who has received surgery for a spindle cell sarcoma, had quite extensive surgery, but then unfortunately developed small volume lung metastases. And in June of last year, the disease progressed, both in the lung and in a right tibial metastasis. And he received palliative radiation to the metastasis in the leg, and then 2 cycles of doxorubicin and ifosfamide. But unfortunately, the cancer progressed. And in September of last year, he started treatment with larotrectinib. And he's had a very durable response to treatment. You can see that the tumors really just shrunk away. And again, it's corresponded with a huge improvement in his quality of life. So prior to starting treatment, he was very symptomatic, felt very tired, had shortness of breath, just generally felt unwell, but now is back at work and enjoying life with his family. I mentioned EZH2 inhibitors in epithelioid sarcoma. Epithelioid sarcoma is a very rare type of sarcoma, accounting for less than 1% of sarcomas, and it's characterized by loss of iron I1, which basically creates a oncogenic dependency or a cancer-driving dependency on something called EZH2. And this is the rationale for using an inhibitor of EZH2 in epithelioid sarcoma. And there could be a rationale for using this type of drug in other sarcomas with iron I1 loss. So there was we did a phase 2 trial of tazemetostat, an oral EZH2 inhibitor in a variety of different cancers. So there were a number of different cohorts. Epithelioid sarcoma was one of the cohorts, and the primary endpoint was the response rate to treatment. So these are the demographic characteristics of the patients recruited into this trial. Again, very typical for this particular histological subtype. The response rate was about 15%, which may not seem very high, but a number of patients with stable disease derived durable benefit from this agent. And this, in combination with the good tolerability of this drug, has led to the FDA approval of this agent. I mentioned tenosynovial giant cell tumor earlier on. There's a good rationale for using CSF1 inhibitors in this disease, and a randomized phase 3 trial has been actually performed in this very rare disease, which showed a significant improvement in response rate over placebo. So in terms of the standard response evaluation criteria, RESIST, for pexidartinib, the response rate was 39% versus 0% for placebo. In general, this agent was relatively well tolerated, but there were serious non-fatal liver toxicity with increased bilirubin in 4% of patients. And notably, there was improved patient symptoms and function on pexidartinib. And that's the slide showing the response rates to the drug in this randomized trial. In desmoid tumor, a number of randomized trials have also been performed. So Dr. Gounder and colleagues performed a randomized phase 3 trial of sorafenib versus placebo. And again, the response rate was the primary endpoint of this trial. And it basically showed a higher response rate in the sorafenib arm. And in terms of the primary endpoint, median progression-free survival, it was not reached for sorafenib, but was 11.3 months for placebo. But I suppose the really interesting thing about this is that the response rate in the placebo arm was 20%, again showing the unpredictable nature of this particular tumor type. So there are a number of other exciting drugs in development. So for chondrosarcoma, there is a drug that is activates the death receptor 5, i.e. basically inducing the death of cancer cells. This has been evaluated in an early stage trial to assess the best dose and schedule for this drug. And a number of chondrosarcoma patients were recruited into this trial. And of note, there were very good partial responses to treatment in 2 patients. And a number of patients also had tumor shrinkage. And these slides show some of these responses. And this has actually led to a randomized phase 2 trial of this drug in advanced chondrosarcoma, which is currently recruiting. Synovial sarcoma, so a number of interesting approaches in this particular type of sarcoma. Dr. Van Tine presented the results of a randomized trial of enlotinib at the ASCO meeting last year. Treatments targeting NY-ESO and other cancer testis antigens have been evaluated in synovial sarcoma and myxoid liposarcoma. And there are a number of other approaches, including BRD9 degraders in synovial sarcoma. So I think Dr. Reichardt may mention and discuss some of these trials in his session on liposarcomas, but there is a rationale for targeting so-called or specific cancer testis antigens in synovial and myxoid liposarcoma, particularly MAGE-A4 and NY-ESO1. And Dr. D'Angelo and colleagues have performed a trial of this type of T-cell therapy in synovial and myxoid liposarcoma. It's important to note that this is a very specific group of patients that are eligible for this trial. They have to be screened for HLA status and also for the antigen or the protein that appears on the cancer cells, so MAGE-A4 in this case. And this slide outlines the schedule for the trial and the treatment. So, as you can see, quite a complex schedule involving chemotherapy, lymphodepletion, followed by T-cell infusion, and then follow up thereafter. And just to give you an idea of the very sort of precision medicine approach that this involves, in this trial, they screened 330 patients for HLA-2. 176 patients had the right HLA type, and of these 176, 106 were MAGE-A4 positive, and 59 patients were enrolled and had their blood taken to grow the T cells in the laboratory, and then 37 went on to receive the T cell therapy. And this included 32 patients with synovial sarcoma. So for synovial sarcoma, the objective response rate was 41%, so 12 out of 29. And there were 2 patients that had a complete response radiologically. So a very impressive result. Of course, there are a number of potential side effects with this approach, particularly the release of cytokines. But in this trial, the so-called cytokine release syndrome was mainly low grade, and patients tolerated this treatment relatively well. But as I mentioned, this is quite a complex process, and it's a very niche approach in terms of identifying almost subgroups of subtypes. So there is a compelling scientific rationale for this precision medicine approach, but we clearly need to have a lot of infrastructure in place to do such a trial. And I suppose one of the major questions, again, is that there were durable responses observed, but how can we identify people that are most likely to benefit from this approach, given that it is so expensive in a way and requires so much infrastructure? The lymphodepletion schedule that's given before the infusion of the T cells does contain an alkylating agent. This could potentially contribute to the efficacy of this approach. We know that ifosfamide rechallenge has activity in synovial sarcoma in particular. And of course, experience is required in managing the toxicity of these approaches, even though in this trial, the cytokine release syndrome was mainly low grade. So Dr. Van Tijn and colleagues performed a phase 3 trial of anlotinib versus dacarbazine in advanced synovial sarcoma. The trial met its primary endpoint in terms of progression-free survival with a hazard ratio of 0.449. So the median progression-free survival was 2.89 months for anlotinib and 1.64 months for dacarbazine. And again, notable to see that a number of people did really well and had very durable benefit from this drug. So again, it's good that we got prospective randomized phase 3 data in advanced synovial sarcoma. But again, one of the major questions remains, how can we identify people most likely to benefit from this particular treatment? And one of the things that we could consider is using this treatment in combination with other drugs in view of its side effect profile. And then finally, just a quick word regarding immunotherapy. The SARC group performed a trial of single agent pembrolizumab with an approximately 18% response rate in advanced sarcomas. Efficacy noted potentially in undifferentiated pleomorphic sarcoma and dedifferentiated liposarcomas. And of course, there are a number of combination therapy trials ongoing, including the Immunosarc trial with Dr. Martin Broto of sunitinib and nivolumab. again, promising activity of this combination approach, particularly in certain subtypes such as alveolar soft part sarcoma. Dr. Wilkie and colleagues have performed a similar trial of axitinib and pembrolizumab. Again, activity noted in alveolar soft part sarcoma in particular. We've performed a trial of gemcitabine and pembrolizumab in leiomyosarcoma and undifferentiated pleomorphic sarcoma. We've seen benefit in pleomorphic sarcoma in terms of responses, but no responses in leiomyosarcoma. So in conclusion, I think there are definitely more systemic therapy options available for advanced disease. There are lots of promising agents in development in clinical trials, but we clearly need a better understanding of the underlying biology of a lot of sarcoma subtypes and incorporate translational studies into clinical trial design. So I'd like to thank all of my colleagues and patients at the Royal Marsden. And with that, I'll say thank you. I've probably said too much and gone on for too long, so I'd welcome any questions.
That was excellent. You really gave us a terrific overview of the update of the medical research landscape in soft tissue sarcoma. We open it now for some questions. Any questions in the room?
I'm pleased that my friend Peter Reichhardt is there, so I'd welcome a discussion. Yes, good to see you, and Roger's there as well, so I think Roger's got a...
Yes, I've learned that.
Yeah.
Okay, my name is Andrea Bolzicco from Orchestra per la Vita, Italy, and we are fighting for epithelial sarcoma, against epithelial sarcoma. I have a couple of questions for Robin. First one is regarding BRD9 degraders. If I will learn that this type of degraders should be applicable also to SMARCB1 negative cancers. So I would like to know if it could be applicable to epithelial sarcoma. That's the first question.
Yeah, thank you. That's an excellent question. I think there is definitely a role for all of these, or a potential role for all of these type of epigenetic agents in certain sarcoma subtypes. I think with regard to epithelioid sarcoma, right now, I think we don't have enough information to be conclusive to say, well, this type of agent will work in epithelioid sarcoma. But I think it would be interesting to explore the use of these agents in epithelioid sarcoma, but I don't think we've got any sort of conclusive evidence right now that they will for sure be effective, if that answers your question.
Okay, thank you very much. Because I've been informed that a new BRD9 degrader has been approved in the USA, and a clinical trial is starting in the USA.
Yeah, no, there are definitely clinical trials starting, and definitely clinical trials starting in synovial sarcoma. But I suppose, you know, even with this sort of how shall I put it, with equipoise, I suppose the whole point of the trial is to see whether the you know, this type of agent will actually work in synovial sarcoma, because even though the rationale is very, very promising, I think one of the things regarding a lot of the epigenetics in synovial sarcoma in particular, and in epithelioid sarcoma, is that it is quite complex, and I think we're just sort of scratching the surface in terms of understanding the biology. I think the results with tazemetostat in epithelioid sarcoma in a way show that it clearly has a very very compelling biological rationale for the use of EZH2 inhibitors in epithelioid sarcoma. But as I mentioned, the response rates and wasn't as high as we would have hoped. And there's clearly more to it than just just one aspect of the epigenetics, if that makes sense. And I suppose that's also the point regarding BRD9 degraders in synovial sarcoma. There's so much that we need to understand regarding not only the mechanisms of response, but also the mechanisms of resistance. And Peter and Denise have probably heard me say this before. I suppose even with older drugs like doxorubicin, there's so much that we don't understand in terms of why some people do derive considerable benefit from doxorubicin and others don't.
I have a second question.
Good. No, I like.
One more question. Okay, you mentioned proteomic data. We are working together with an Italian and another university in the field of immunotherapy. We would like to move, and we hope to move quickly to a study with mice, but there is a lack of proteomic data regarding epithelioid sarcoma. You mentioned some studies regarding proteomic data. Are you working on that?
Yes, so that was in the context of the accelerator grant in localized sarcomas. So that particular grant focuses on 5, I suppose, more common subtypes. So dedifferentiated liposarcoma, synovial sarcoma, malignant peripheral nerve sheath tumor, and vascular sarcomas. We do have some data regarding epithelioid sarcoma, but that's mainly in the metastatic setting, and maybe we could connect after the meeting regarding that, because I think for such a rare subtype of sarcoma, it would be good to consider working together on that. These data haven't been published yet, so maybe good if we connected after the meeting, if that's possible.
Okay, perfect. Thank you very much.
Yeah, no, thank you.
Dr. Reichert?
Edition. Hi.
Hi.
Robin. Good to
How
see
are
you, even if it's just virtual. Just in addition what you said, I think it needs to be made clear that tazemetostat, you mentioned, is licensed in the US by the FDA. It is not in Europe. The EMA has not accepted that. The same applies to pexidartinib. So there are still differences between the US opinion and the European opinion. The European opinion is a little bit more toxicity concerned. So they normally ask for more benefit to justify a given amount of toxicity. So these two agents that were excellently mentioned by Robin are available in the US, but not in Europe, just for the completeness of that.
Yeah, I think that's a great point, Peter. And of course, even I suppose pazopanib it is not funded in the UK, so there can be variation, unfortunately, in the access of drugs, which is almost a discussion in itself, really.
So, Robin, you had mentioned about how important it is that we understand better the biology, and it makes me think about the collection of biospecimens and the important correlatives and the role that patients can play in that. Do you see that those are being incorporated more into the trials, and are you actually getting the samples you need to answer these really important questions after you conduct these trials?
Yeah, I think that, again, that's a great question, Denise. I think it is definitely improving in that respect. And I think collaboration is very important. But of course, one of the things to remember is that there's a lot of bureaucracy and a lot of regulation to sort of not get through, but to work through in terms of setting up those sort of trials. And clearly they're more expensive to run. But given the fact that there have been a number of negative phase 3 trials, particularly in the first line setting for advanced soft tissue sarcomas over the last 10 years. And as I mentioned, we still don't really know why some tumors respond so well to doxorubicin. We don't really know why those trials were negative, even though a number of patients that were treated on the experimental arms or the combination arms of those trials derived benefit from being treated with those newer drugs under evaluation. And I think the only way we're going to get a better understanding of that is to sequential biopsies, particularly in earlier stage trials. And that sort of follows on from the excellent question regarding the use of BRD9 degraders, the use of tazemetostat in synovial sarcoma and epithelioid sarcoma. Why did some patients on the phase 2 trial of tazemetostat derive such durable benefit, whereas other people unfortunately didn't? And I suppose the same applies to the synovial sarcoma cohort of the tazemetostat trial, in that the results for that cohort were relatively disappointing, despite there being a very reasonable biological rationale for using the drug in synovial sarcomas. So I think the short answer to your question, Denise, is that I think things are getting better. More trials are incorporating correlative studies, but given the complexity and the heterogeneity of sarcomas in general, it's still there's still a lot of work to do.
Absolutely. We're going to take 2 last questions. Roger, I'll go to you, and then Annika.
Thank you, Denise. Robin, it's really a comment rather than anything else. I'm very cautious about long-term analysis of the RTC 62931 because it dates from a time when there was no central pathology review. And I know from my own experience, I was treated on 62092, which ran at about the same time. I was originally misdiagnosed and I'm reported in 62092 incorrectly. I raised this with Martine van Glabacker a long time ago. It has to be a long time ago. And she said, categorically, Roger, we will not republish. But I just want to caution anything involving histology and trials of that age.
Yeah, I think as always, Roger, that's a fantastic point. I think it's very difficult. It's just to almost put things in context regarding the use of neoadjuvant chemotherapy. It's such a controversial topic, but I do think that despite all the limitations, putting all of this, in a way, new information, new analyses together, it's very interesting and very, very thought-provoking. So I think it's not just one aspect, i.e. Dr. Pasquale's reanalysis of that trial, but it's also the fact that you can put that together with, for instance, the PASARC predictor as well, which is again another fantastic study by the Dutch team, and the randomized trial conducted by Alex Cronky and colleagues. So I think you're absolutely right, but it's to try and just put everything in context. And yeah. I suppose, in a way, to have a definitive trial of chemotherapy versus no chemotherapy for localized disease, we'd almost have to rewind the clock to 1987 or something, because it's almost become such a controversial topic. I think it would be very difficult to do that sort of trial now.
I agree with you. The data we do have would suggest that if nothing else, the ethical questions that would be raised are huge, but of course there are big scientific questions too.
Indeed.
Thank you, Robin. I take your point.
Yeah, good to see you. You and I had the rail strikes to contend with. I think Sorrel as well.
Well, for me, it just went quiet because we can distantly hear the railway at home, and we've had quiet.
This is Annika from Finland. I don't know if you remember me, Robin.
Yes, I do. How are you?
Fine, thank you. I have a kind of a general question about the research in sarcoma, as I have the educated guess that my husband's mesenchymal chondrosarcoma was mitochondrial disease in some way. So have you seen some research about sarcomas and mitochondrial dysfunction?
Off the top of my head, I'm just trying to think. I'd need to look into that, but I have nothing that I have seen recently. But I'll certainly have a look at that, and that's a very interesting question.
Well, thank you very much, Professor
Yeah,
Jones, for
maybe I
your
can
time
get...
today and for your excellent presentation and for the discussion. Thank you so much. And now we turn to you, Dr. Reichart, to certainly give applause.